right, we saved the best part of the pump for last.
Whoa, no pressure.
So this session's about the active transport, the migrant pump. And our first speaker is Dr. Svenja Halfter. And Svenja is from NEWA, the National Institute for Water and Atmospheric Research in New Zealand. And she is an institutional postdoctoral fellow there. Svenja's interests are in zooplankton ecology and biogeochemistry. And she's worked a lot, especially in the Southern Ocean. And she is importantly the leader of our active flux working group in the JETZON project. So she's been thinking a lot about this lately, and she's put together a really nice talk on sort of our progress so far. So take it away, Svenja.
I put together a talk, not a nice one, but a talk. And there's some slides here. First I want to probably echo Adrian's point that, have had a blast. This was an amazing opportunity to talk to so many interested people, and, it's just great to be here. My first time in the US, so I'm really excited. Culturally interesting. What's up with your toilets? You have so much water in there. What's happening? Anyway, well, apart from that, it has been a really good time. It's really just flushing. It's great. Anyway, what I want to talk about is, if I remember correctly, is the active flux. And there is a bit of overlap from last week. So you've seen all the beautiful visuals and all the beautiful fish and boar. So I have some pictures, but not as many beautiful ones as last week. But we'll talk more about numbers and at least a few numbers I found. So anyway, but this is of course not my own work alone. I'm, supported by a big working group who read a few papers for me, which is great. And we did a bit over a long time, and then basically everything over the last 2 weeks. So I'm happy I have slides here, and I'm here now for you talking. And all those amazing people have been really great. But if you want to join the group, and if you have other ideas, just following up on my talk, just contact me, send me an email, and I add you on. We'll be good. And also, especially a thank you to Debbie, who's been a great mentor so far. It's just amazing that she went from reviewing my thesis to now collaborating. It's amazing. It's, I think, just, the power of the community. So what do I want to talk about today? I realized I don't have any fish. I'm sorry, but it's okay, I guess. I guess that's it. Thank you so much, everybody. I think I pressed the wrong button. How do I unpress it? Everything is down. That's a problem. Okay. Let's continue. All right. I will talk about, introduce how migrating organisms impact the downward carbon export. Then I will talk a bit, just a slide about the review process, what we went through mentally. Then I talk about numbers, all the magnitudes, carbon export globally and different regions, and then also through the water column. At least there's one graph. Then uncertainties and drivers, and apologies again, there will be lots of overlap from last week, but some people haven't been here last week, so I guess that's good as a repetition. And there might be a pop quiz later, so I can't promise anything here. And then suggestions for next steps in our literature review work, but also in future, where we want to go with that. As an introduction, you've seen this before a few times now, but I will focus on the two major pathways that migrating organisms inject carbon into the ocean. And that's really this thing between the passive flux. So all zooplankton and fish, they poo a lot, so they contribute to the passive flux too. But we're really focusing on the active injection of carbon flux to deeper depths. And this is the mesopelagic migrant pump and the seasonal lipid pump, and I'll explain what they both mean in a second. And here they are. So Phil made a nice graphic. I guess he didn't make it himself, but anyway, nice graphic in his paper. So what is the mesopelagic migrant pump? So we have, I guess you all know that animals will migrate up during the night to feed for example, there we go, to feed at the surface, and then they would migrate down to deeper depths. And then poo, for example, here's a sexy particle for Chloe, where they actively inject carbon in deeper layers. And their forms are, they poo, so they produce bigger pellets, and caucuses, so it's PSC that we inject. It's DACs, or they respire, of course, but they're, they're based on their, what they fed at the surface. They excrete DOC, and also, well, we don't really talk about it, but also if they die, we also have shells and ocellates that actually, go further down, but we don't really talk about that. So I just want to mention it here. And here's one example. There's a nice study that already reviewed a few active flux contributions by, I think, micronectin mostly. And you can easily see here that there's different magnitudes, and they really differ between gears. So that's the main problem. I guess it's the main problem, or the main also difference to the passive flux, that you have a sediment trap in the water for the passive flux. You get a cup of dirt, then you measure the dirt, and you have the measurement. Whereas for the active flux, we observe the biomass with different gears that are all biased, and then from there we calculate and estimate and assume a lot of rates, and then the carbon export. So that's why it's always indirect how we measure that, and that's, I think, making it more complicated. And as you also can see, that there are huge spatial and temporal gaps. So we are pretty well covered in the North and Central Atlantic, but for example, Indian sector or Indian Ocean, it's nothing really on the east of this study. And then the seasonal lipid pump is again migrating organisms, but this is kind of an overwintering strategy. So it's really well studied for the Northern Hemisphere. So they're polar copepods, and they feed in the summer season on what's in the water, phytoplankton mostly, and they build up these big lipid reserves. So they're really fat and chubby and happy. And then they migrate down, and it is quite deep. So these depths are, depending on the species and the respiration, but they go quite deep. So they actively inject carbon at that depth because during the time they spend in dormancy usually, they will either reproduce at depth, or so they die, just sinking down, or they reproduce. Oh no, they come up again partially and then reproduce and then die, or there's like a lot of life cycles. But what's important here that they respire. Sorry, going back. Yeah, the caucuses are important. Then they respire what they have, the lipid reserves. And then, well, we don't really know about either fecal pellets or excretion because when we assume they're dormant, so they don't really feed, then that's not irrelevant. But, we don't really know what's happening down there. So I still added those pathways here. Yeah. And here's an example, and this is the, one part that's really well studied because we have so big copepods in the North Atlantic that are vertical migrating. Overwintering at depth. So we have colonists from Marshall, for example, and of course others. And we can really see here in comparison to, is there a pointer maybe? In comparison to passive flux that are interacting with the nitrogen cycle and the carbon cycle, it sinks down and has an influence on the nutrient concentration at surface. They migrate down and they're just respiring their lipids. So they're really just shunting down lipids, and that's why it's also called lipid pump. Makes sense. I thought about it. And here you can see here, it's really dependent on where they're distributed and how many are in the water column and how many migrate down that they impact the carbon flux. And here we go. This is the methods. We have a literature review we did, and I used a template that got bigger and bigger over time for some reason because we were interested in all the data. And this was me when I started, and this is me now. So anyway. It's all right. We took out a lot of literature, information from the literature. So information of the publication, of course, study region, study time, environmental parameters such as chlorophyll A concentration, MPP, mixed layer depth, and all kind of things we thought could be interesting. Oxygen, all the things. Study methods, which gear has been used, because that's, as we just seen another example, it's quite important. The migrator biomass and carbon standing stock, the carbon flux, obviously, And then, uncertainties and potential drivers, what been mentioned in the literature, because, as I said, because it's the indirect methods, we go from, biased view on the biomass to export, which is, a lot. So, and you've seen this paper, I don't know, cited a lot of times already, but I will just follow up because it's a really nice paper, I think. And it shows here the microgrid pump, the importance of the microgrid pump compared to other export processes of PLC. You really see that it's, global difference. Some have really few. It's not really important here in the subtropical regions, but more important in the upwelling regions, North Atlantic, and of course, a bit in the Southern Ocean here. So yeah. And I assume you have seen this number already a few times now, but sorry to repeat it. In the end, you will take this away, right? So this is the 10%, you remember, 10 petagram per carbon per year, we assume for the whole biological carbon pump right now, of which 70% is the gravitational flux, also mainly dominated by zooplankton, it's called physical processes, 20%, and then 10% is zooplankton migrations, ended up around 1 petagram per year. So it doesn't sound like much, but, because they, go quite deep or go, actively inject carbon to a deeper depth, they will stay there, 150 years. So it's quite important if you look at those timescales and sequestration times. Here's another paper by Phil et al. And here we go. And I just marked the MMP and the SLPs in green and blue. And you can really see here that all the export rates for MMP are a bit higher. Oh, sorry, start with the SLP because it didn't make sense. Here we go. So the SLP is kind of between 0.25 and 1 petagram per year. They assume 50% in the North Atlantic, and the MMP is kind of a bit higher in the range. You can see here in the graphic that it is a bit more. And then they have also another number, which is like 1 petagram per year. So there's different numbers they reviewed and came up with. But as you can see here again, there's a long sequestration time because, especially the SLP, they go so deep down. It's basically shunting down carbon to 2,000-meter depth. That's pretty significant. And then 2 more papers. I'm sorry, I think that goes through a few numbers here. One is by Pinty et al., who looked at 5 seasonal migrating copepod species, and they all do this vertical migrating seasonally. And you can see here, they all have really different. Oh shit! Sorry. Spoiler. They have really different life cycles of Schematicos. It's a polar or subpolar species. Ababoreus is a polar species up in the northern hemisphere. Colonides acutus, southern ocean. Colonides anatalis in more upwelling regions, Angola, Karen, and Arabian Sea. And Neconos tonsus is what I usually work with in the Southern Ocean. You can see here they're all migrating and they're all doing their thing, injecting carbon. But because they're so different, how do you want to put this in models? I guess that's probably the main question here. And this is just 5 species. There's so many copepod species that are seasonal migrating, but we don't really consider them at all. So huge gap, what do you say? And they come up with a quite low number in this case. But if you think about it, this is at 1,000-meter depth. So, depends on how you want to see it. At surface, this would be like really low, but because it's at 1,000-meter depth, you have to really consider it. And Record et al., they use different models, and they compare different models to model the global lipidscape. So they looked at different traits. How likely is the copepods, or do they have the trait to seasonal migrate downwards? And they used, in the end, came up with I thought the life history model would be best, and it came up with a bit higher number than Pinty et al., but also kind of in the range. And here I just plotted some of those numbers, but also a few others. And if you, on the left side, you can see the MMP in pink, green, or pink, red, or something. And then the SLP, we have 3 values here for global numbers. So this is all the data we have. How nice. And then, if you look at the different pathways, when I looked at the different papers, I had everybody taking out which pathways they considered was quite different between studies sometimes. So for the MMP, they usually used fecal pellet production, gut flux, and digestion, which was really, it's not quite the same, but kind of. So I used to see it interchangeably, but, we have to really look into the different definitions, how you define those great, really. They used often also respiration, which is great, but then carcass flux and DOC, even though they might be important, not really well considered in literature. And then we look at SLP, again, a good representation of respiration because we know they respire the lipid reserves. But only, and carcass flux is also considered sometimes. But then for others, we rarely can consider egg release, even though some of the copepods reproduce at depth and then the eggs would come up. So we would have to subtract that from, what we're putting down. So it's not really well considered. But also in reality, I put them all in one graph here, and it's nice, right? So if you just look at that graph, you're like, oh, actually, should we just focus on the MMP because the variability is higher and it looks like it's a bit more important than SLP. Again, this is 1,000-meter depth, and this is up at 200, 500 meters. So different. Then Saba et al. Only considered fish. Finty, though, as I said, only 5 species of copepods are incomplete. So we really not even comparing apples and raspberries and lemons. We really have a whole fruit bowl out there. We're comparing stuff with. And it's really, we always pretend they're comparable, but we really have to go back to the papers and then see what they're actually putting in, how they're assuming things. And it's not bad, because there's probably different ways to get to that number, but, we should definitely keep that in mind. You've seen this a few thousand times now. It's Adrian's fault, and and I put them up here, but I haven't really used those provinces yet because we just have not really a lot of data points. So, for example, the mesopelagic migrant pump, you can see here, it's actually not too bad of a spread. If you wonder what happened here, well, I think Santiago happened, and who single-handedly tried to solve the whole problem by himself. But you can see it, there's a lot of different points, but there's still a few data points missing because we haven't quite finished the review yet. There's many papers that focus on organism-mediated flux, but mostly on the passive side of things, and sometimes they not even distinguish between passive and active flux. So it's really hard to get numbers out of papers sometimes. And then many papers only focus on single groups or single organisms even, or single species. So it's really hard to compare, if you have magnitude in the end, really hard to compare those different values. And then also there's different taxonomic resolution considered. Sometimes they just bulk a number together and call it zooplankton. Someone say micronectins, sometimes fish, but then there's a lot of overlap between those definitions as well. So sometimes it's hard to directly compare these values we get. And here I plotted those over latitude, and this is not complete yet. I thought I'd just see if there's a nice pattern. It's probably a good representation in the northern hemisphere right now, over latitude, but southern hemisphere, you can see there's a huge gap at around, here in the lower latitudes of the southern hemisphere. So we have huge gaps, and currently I don't see really a pattern. And I also through the water column, I wanted to put a curve through, but then I thought, this is actually, we just bulk everything together. I'm not sure if that's actually making sense in a scientific way. So you see here, but people will report at, the different depths, but there's higher flux at the surface, I guess, or closer to the surface, and then a bit declining, but we also have really high values here. So it's really just the start of the data analysis. I just thought I'd show you some plots, so you don't think I'm lazy. And here we go. And there's the seasonal lipid pump. As I said, it's really focused on the North Atlantic because there have been, quite a few people working on copepods there, and we have really these big, chunky copepods as well. You can actually see with your bare eyes. But you can see, I just put up the different species they're used, and if you think, they look all the same, well, welcome to my life. But you really see that, we have maybe one data point in the Indian sector, so we definitely need to, see if there's some other vertical migrating copepods there, and I think there are. And there's some underrepresentation of other groups to other regions too. So it's really just, we really don't consider all the data right now, or even all the copepods that could seasonal migrate. And again, by species, you can see the same pattern. Lots of stuff is happening on the. Here we go. Lots of stuff is happening on the northern hemisphere. And again, we have these huge gaps. And currently, there's a bit of a decline here, but I mean, we have like 4 data points here. Like, you know, I don't wanna like be overestimating the whole thing right now. So this is still in progress. And as you can see, I didn't mention it, but there's different units, and I left them at their units right now. This is gram carbon per year, and whereas the other MMP, I have milligram carbon per day. And obviously, the seasonal lipid pump works on the seasonal scale, makes sense. And then, you know, the seasonal lipid pump works in a seasonal scale. So even combining and finding one unit is quite hard. It's not a mathematical problem. I can divide by the days they're down there, but how many days are down there? You know, do I introduce another bias by assuming a certain number of days because I then actually don't know when they migrate down and when they migrate up? Yeah, and then ICP high in the Southern Ocean. Well, there are different ways to calculate that. So we just hopefully add some more data points. You know, then there's lack of data, and especially distribution data, because from distribution data we were able to calculate carbon export, but you know, because we don't have that. Well, we have that, but we haven't looked at it yet. We know for 200 years that you know, zooplankton and fish are migrating, but this only recently we just started putting numbers to that. And yeah, what about other species? We looked at, you know, 5 or 6 different copperpot species right now, but there's so many out there that might also migrate. And what about lower latitudes? Of course, there's a data gap there. Yeah, so talking about the caveats, there's various carbon units, as I said. It's really hard to compare sometimes. And again, it's not a mathematical problem. It is an ecological problem that I have. I don't want to bulk numbers together that don't belong together. Carbon pumps at different time scales. I already mentioned that. One is seasonal, one is daily. We have different carbon pathways, and they're considered in literature, and each paper kind of has their own way to estimate the different pathways, and it's really confusing if you really go back into the detail. Like, how do you come up with this number? Well, you know, everybody has their own favorite little equations, and we cite each other, but it's a mess. My God. And of course, there's the risk of double accounting. You know, when you know something is pooping up there, let's say, pooping up at the surface, and it sinks down its gravitational flux. Then they migrate down its active flux. But how long? You know, when does it start to be active? And what is if that's, you know, being fed on by or eaten by fish? You know, is that still active? Well, I don't know. I mean, there's so many. It's a bit of the risk of double accounting things. And, you know, here's one slide about current gaps and uncertainties, and I'm sorry if I repeat stuff that has been already said, but here's just a summary, what we took out of the literature. And of course, it's not an exhaustive list. We have to, you know, consider a few more things, but it's just a summary. So there's a huge uncertainty about biomass. We've already talked about this. There are different methodologies. There's a different sampling, and every sampling method we use introduces different biases, and we don't really understand those sometimes. We assume there's different capture efficiency, and we use sometimes factors to correct for that, but all of these factors actually correct. We just assume a lot. There's seasonalities, especially in the polar regions. Almost our information comes from spring and summer campaigns when, you know, there's no sea ice and it's actually accessible. But how do we figure out how deep those copperpots are when we can't be there? Obviously, you know, there are no gliders and things, and then, you know, stuff is happening remotely, but it's still a bit sparse. Metabolic rates is a big one. You know, what's the respiration at depth during diapause? What's the mortality rate? Mortality rate? I mean, yeah, they die, I guess, but how much really? Gut flux, molting, we don't really care because, well, crustacean molts regularly, but we don't know how frequent. How much do they poo at depth? DOC excretion, we use a single factor, usually a percentage of this could be DOC excretion, but is that correct? Yeah. And then not all the carbon pathways are always included in the papers, and it's, yeah, really makes it hard to compare studies. Life cycle, what happens to copperpots after spawning? I guess they die, but maybe they migrate back up and just have another go at it or something. What's the regional cues for diapause? You know, some things about that, but not really much about other species. Some regions are really understudied. There's different methods to calculate the length of diapause, and that's the main problem for the seasonal lipid pump, because just, you know, we have to calculate it. We need to know how long they're down there, how long they're injecting carbon, but, you know, we just, yeah. We're like, 200 days, 300 days, you know. In the deep sea processes, yeah, there's a lot happening down there. Predation, fecal pellets, sinking rays that we don't know much about. If they're able to feed, they produce particles. Is that the sinking flux? Is that or the gravitational flux? Is that the active flux? Yeah, we don't know. Degradation and consumption of carcasses, I guess they will be eaten, but how many of them, depending on the predators community, but who's predating on them? Yeah. Estimation issues, you know, we try to extrapolate based on a few single studies we have, you know, to extrapolate for the whole region. How accurate is that actually? How do we convert biomass to carbon? That's, you know, good, well known for some species, but some we just try to use something from a different group, and I don't know how correct that is. Double accounting, again, variability, it's really variable. These processes are, you know, highly variable on a temporal scale, and seasonal into annual, of course. Spatial between regions, you know, between species, you know, even within populations. Not all of the population would migrate regularly. So, yeah, it's a mess. It's a mess. And then modeling, there's sometimes low resolution and high latitude systems because as an input we put in satellite-derived primary productivity or something, and with the higher latitudes we don't always have satellite images. And then, you know, sometimes they have just really low resolution of regional variability just because we can't put numbers in, which, you know, doesn't resolve well processes. And then as a drivers of active carbon flux, and again, this is not an exhaustive list, this is just what we took out of the literature. You know, for example, there's then all the environmental factors, again, non-exhaustive, with the temperature, which of course affects the metabolism of animals. Primary productivity and food availability, do they always migrate? Maybe, you know, if there's a lot of stuff in the water at the surface, maybe it's worth staying up there, so they're not always migrating. Mixed layer depth, light attenuation, it kind of also defines the depth of the migration. Mesoscale eddies and upwelling and oxygen concentration, you heard about it a bit. And then, of course, there's always things on the organism and population level that drive active carbon flux. Some abundance and community composition, how many actually go down and overwinter at depth, individual diet that kind of formulates their, you know, what kind of phytoplankton they eat is really important, what kind of lipid they form, and lipid composition. DVM behavior, already said, not all of the community would always migrate, really complicated. Individual lipid content, of course, because it defines the length of the diapause that we're able to do, and predation, we don't know much about that. And the next steps for this, of course, first finalize the literature review, because there's still a few papers we have to read, not heaps, but a few. And then look at the importance of environmental factors, because I haven't had time to do that yet, but, you know, and then try to identify drivers of change. And also, you know, because we know that the ocean is changing, we have to understand how it would look in the future. We know that the sampling gear is really important because it's really, you know, everything we use is biased, but how much and how does it affect the final number we get out of it? And then hopefully we kind of end up with something like recommend more consistent ways to estimate MMP and SLP, at least come up with, at least consider the same carbon pathways in every paper, you know, whatever equation they use, but it would be nice to have at least all the, you know, carbon pathways covered. That would be great. And develop some kind of framework to facilitate that. So in summary, the 10% you should remember, migrating organisms contribute to carbon export and sequestration. The magnitude of, however, is really, it's really changing. And it's, of course, less than the gravitational flux, but it's dependent on the variability, and there's currently really large spatial and temporal variability. And we also, yeah, I mean, I'm just repeating what everybody else has said, but we need more empirical data or estimation, local estimation, to just be confident to when we extrapolate things, right? Because right now I feel not confident to extrapolate beyond my little single field study because, yeah, I don't know, it's, you know, not different somewhere else. And then, you know, there's large uncertainty about certain physiological processes, so we can't put a number on it, we can't include it in models. And of course, overall biomass is always you know, a topic, and, you know, we need to know a bit better to integrate them into models. And then, you know, because of those variability and uncertainty, it's currently difficult to understand and project future changes, climate change, but also fisheries and deep sea mining. How does everything, you know, the stresses we put on the system affect the whole thing? So yeah, thank you for an amazing time. And before I close, I will just briefly just I don't know how to say it, advertise our session at the Zooplankton Symposium next year in March in Hobart. We will convene a session about the zooplankton and the biological carbon pump. And if you're working on that topic, please come to submit the abstract until the 30th of September. There's support for ECRs to join the conference. So please come and submit your abstract. Okay, thank you so much. That's it. Questions? Concerns?
Thank you, Sonia. We have time for questions?
Ken Missler, thanks for that. Trying to link this week to last week and have you say a little more about fish versus zooplankton in terms of their contribution. You had the Saba paper there, and you said you couldn't see a difference, but
yeah.
Can you even say, do fish make fecal pellets? I've never seen one in a gel trap. Can we say something about DVM, maybe not in magnitude, but the role they play in terms of what they do when they're down?
Totally. Yeah, no, it's an emerging topic now. I mean, there's a few papers out now that you look at fish contribution because we looked at zooplankton for a long time, and obviously they're still important. But yeah, fish only has been recently considered, well, for export at least. And because they migrate, can migrate quite deep, and their gut flux is a bit lower than, zooplankton, they feed, and then, a few hours later it comes out like, they produce a few fecal pellets and really fast. Whereas the fish, they're like us, I guess. The bigger they are, the longer it takes for food to go down. So when they migrate down, they would inject more carbon because the gut flux is a bit lower. And the fecal pellets are bigger too, so if they sink faster, we would actually contribute more to the carbon flux. But I mean, it depends on how many there are. I mean, that's currently the biggest uncertainty of the biomass of fish.
But has anyone really seen fish fecal pellets? Have we quantified them in the traps? I think of Colleen Durkin's kind of work and others. People are looking pretty hard now at what's in those gravitational flux.
Yeah.
I haven't seen that.
I think so. I think there's a paper out now. I mean, Saba et al. Summarized it a bit, but who was I wish I was remembering names now. But there was a paper out, looking at, they found a lot of fish fecal pellets, and that's what I got thinking about, what about fish, actually, because they're so massive compared to, whatever we see in the ecosystem. So, yeah, I think there's some more papers out now, but, yeah, it's only in starting.
Indeed, that's just starting. We occasionally see something that looks like a fish fecal pellet in a trap, but they're big and they're rare. So, yeah.
I guess the general problem is also that all these migrators, they're not really ending up sometimes in the sediment traps because they're alive and they're not actively sinking in, right? So they would avoid. Some of these seasonal migrating copepods, they're not really found in traps. But we know that because, there's some stuff coming up. So, and now we have actually some evidence. There was a winter voyage now because I'm looking at this specific region in the subarctic Southern Ocean. And we now have a winter cruise, which use a depth stratified sampling. We actually know now where they hang out. And it's like the first time we actually, we just assumed before, but now we actually have some data. They go quite deep. They hang out around 1,000 meters.
Yeah, hi. Thanks for this. I mean, from a modeling point of view, there's not a data set available to test your models. I just asked Debbie for the stuff that we've done. So this is great. There's a couple things that will be really useful for the modeling communities, like knowing or at least verifying what Daniele Bianchi has done in terms of the depth at which they go to, how, we assume there's a magic isolume, and that's modified by what the oxygen is, and, just spending some time with that. The other thing is that all the modeling that I've been involved with to look at DVM, we assume that there's some magic fraction of the zooplankton that migrates, and it would be something that it would be. I think it's something different than 50%, but and we do vary this, but those would be things that would be super useful to have these data sets available for everybody to use, because right now one last thing.
Yeah.
You mentioned about the issue of not everything's measured, not every pathway is measured when you have Is there a way to, Pete, is there a way to put the DOC flux in terms of the respiration flux and to parameterize it that way so that we can get better, more, is that something that's being considered?
I hopefully, I mean, to be realistic, we would never figure out everything. Every little rate we have to know. We have to assume at some point, or just, I think, figuring out the best educated guess and guesstimate sometimes, and figuring out, okay, this is the most realistic thing we have right now. And, you know, in the end, we will never figure out everything, you know, what's happening at depth, you know, maybe in 20 years. But, I mean, it's good. We have work for the next 20 years. It's nice. But, yeah, we will never figure out everything. I think we have to get to a point where we cheat enough to get away with it.
And I would just add, I think we can cheat. I think we know enough about, I think we know enough about the physiology that we can say, that, you know, this much, if given this much respiration, about this much is going to DOC, that there's variability, but we have bounds on that. So, and it strikes me we could do that in our spreadsheet where some of those are missing, we can add them in to get a good estimate. And it also strikes me we should probably have another column for fraction of migrating biomass. Those are, we could do that. Thank you for that suggestion.
Totally.
Yeah.
Good. Oh, yes. We want all the things.
Any other questions for Svenja? Okay, great. I think you're going to introduce our
awesome. Yeah.
other speakers.
Cool. Now,
okay.
Let's move on to, thank you.